So this video is part of a two-part series where we are going to be looking at, first, how do we go about isolating autoinducers? And secondly, once we have isolated an autoinducer, what are some of the chemical structures that are common amongst these types of molecules? Remember from the last video that autoinducers are the molecules that are involved in quorum sensing, where quorum sensing is that phenomenon where single-celled organisms can behave in a coordinated way, much like they are multicellular organisms, with the method through which that works being that the cells produce these autoinducers constantly, and when the cells are at low concentration, the autoinducer will be at low concentration. On the other hand, once the concentration of bacteria in a particular environment increases, the concentration of the autoinducer will increase as well, resulting in a switch or a transition in the behavior of the bacterial population to become more virulent, to form biofilms, to bioluminesce, or a variety of other things. How in the world would a scientist go about discovering a new autoinducer molecule? We would begin by making an observation and a hypothesis about a particular behavior that was controlled by the process of quorum sensing. For example, we could hypothesize that bioluminescence, the emission of light by bacteria, a particular bacterium under study, is controlled by quorum sensing. And when we say it's controlled by quorum sensing, what we're saying is that the bacteria are releasing some small molecule autoinducer, and once that autoinducer reaches a certain concentration, it triggers the bacteria to begin emitting light. And so if we were to test this hypothesis experimentally and aim to identify the particular compound or compounds that were responsible for this quorum sensing behavior, what we could do is we could grow up cultures of bacteria, and I'm just going to show an Erlenmeyer flask here, which is a common type of vessel in which to grow bacteria, and we could have the bacteria growing in a liquid nutrient growth bath. So highlighting here in yellow, the mixture of nutrients that are present there that enabled the bacteria to grow and divide. And so what would happen within this flask is that we could leave that flask growing until a high concentration of bacteria were achieved and bioluminescence was noted. So we started to notice that suddenly the bacteria culture was glowing in the dark once it reached that certain high concentration of bacteria. And assuming or hypothesizing that bioluminescence that's been triggered was triggered by a quorum sensing molecule, we could then aim to isolate and identify that quorum sensing molecule, the autoinducer in other words, from this flask of bacteria. And so in order to accomplish that, what one could do is extract the organic molecules from that culture flask of bacteria. So this would be a really complex mixture of compounds, much like when we were looking in the metabolomics unit, we talked about how organisms will typically produce in their metabolome thousands of different molecules. And so we would extract those organic molecules using a technique such as liquid-liquid extraction in a separatory funnel to separate the organic molecules into an organic solvent such as ethyl acetate, leaving the aqueous materials and the aqueous soluble compounds behind. So we'd extract that suite of organic molecules to generate what we could just refer to as the chemical extract or the crude chemical extract. Assuming that chemical extract does indeed contain a quorum sensing molecule, an autoinducer, what we could do is take that chemical extract, that mixture of different compounds produced by the organism, we could supplement non-dense, low abundance, low concentration bacteria cultures with this chemical extract So in other words, we could take a flask that contained a very low concentration of the bacterium of interest, and into that flask also deposit the chemical extract, and then we could monitor the emission of light. In other words, the bioluminescence could be monitored. And assuming that our treatment samples that were supplemented with the chemical extract emitted light in greater quantities than control cultures that were lacking chemical extract, that would indicate to us that that chemical extract contained an autoinducer. So in other words, we would be emulating, by virtue of adding that chemical extract to our low concentration bacterial culture, we would be emulating that the bacteria were present at high concentration because we have a high concentration of the autoinducer chemical. And so assuming that we observed that our treatment cultures that were subject to the chemical extract did indeed emit light, that would indicate to us that the chemical extract contained a autoinducer molecule. And so from there, the task would become, how do we go about identifying that particular autoinducer molecule? And a common way that is approached to do this is a technique called bioassay-guided fractionation. So when we say bioassay, what we're referring to is some test of the biological function of compounds within that mixture. So in this case, the bioassay would be the evaluation of the change in emission of light or bioluminescence in response to the chemical extract. So this would be our bioassay, formed by incubating cells with the chemical extract and monitoring the emission of light and how that emission of light changes for treatments that have been exposed to the chemical extract relative to the controls that have not. So with the bioassay guided fractionation, adding that term fractionation on there, what is done is that the chemical extract is separated into multiple fractions based on some physical property of the molecules that are contained within that chemical extract mixture. So an example of that would be separating the compounds in the mixture based on column chromatography, where a column is packed with a stationary phase being a solid substrate that is going to remain stationary during the experiment. So this would be our stationary phase. A common stationary phase that will separate compounds based on polarity is silica. So silica gel packed into that column will enable the compounds that are applied to that column to be separated based on polarity. And so the way that this would work is one would have a series of test tubes lined up. And as the sample was loaded to the top of the column here, our chemical mixture, the chemical extract, a series of solvents differing in polarity would be added to the top of the column. So solvent would be applied here, also known as the mobile phase because it's going to be moving through. And what would happen is that that mobile phase would carry the chemical extract compounds through the column going toward the outspout here, and the compounds are going to travel at different rates through this stationary phase based on their polarities. And so by collecting fractions at different time points as this elution takes place, as the compounds move through the column, what we do is generate a series of fractions of different polarities, where I'm representing these different fractions and the different compounds contained in each fraction by coloring in the test tubes with different colors here. So we would generally generate somewhere between 4 and up to 40 or 50 different fractions as a result of this column chromatography experiment. Then at that point, we would have no idea which of these particular fractions contained the autoinducer molecule. And so in order to determine which of those fractions contains the autoinducer molecule, we would take this a step further getting into that part about this being bioassay-guided fractionation, and we would test each of those fractions individually for its ability to cause the bacteria to emit light. And now chances are here, at this stage of the game, if we have started from a chemical extract that contains hundreds or thousands of different compounds, and we've done one round of column chromatography, generating several fractions, generally less than 50 fractions, certainly, and we test each of those fractions will still contain multiple compounds. In other words, the fraction will contain a mixture of compounds of a certain polarity range. And so by testing each of those fractions for its ability to cause bacterial light emission, what we can do is narrow in on one or two particular fractions that elicit this biological response of triggering the bacteria to emit light. So what we can do is narrow down the so-called active fraction. And for example, in this case, if we say we had these 4 fractions here, and we evaluated all 4 of those in our bioassay, we found that only this 4th fraction, the one that has our red compound, elicited the emission of light. The others, when supplemented into the bacterial cultures, caused no change in the emission of light. We can narrow down that this fraction contains the autoinducer compound. And so from there, what one would do is additional round or rounds of separation of fractionation. And generally that could be by things like additional rounds of column chromatography on a lab benchtop, or using high-performance liquid chromatography, which generally allows higher resolution separation of the compounds from a mixture than is possible using a benchtop column that is run manually. So through additional rounds of fractionation with HPLC, one would generate additional fractions, which I'm illustrating my little test tubes here. And in those fractions, we would have mixtures of compounds that were more simplified than in the prior round of chromatography, because as we get further and further along here, we're getting closer and closer to a pure compound. We would then take those fractions that we generated and test each of those fractions in our bioassay, and continue doing that as needed, repeating some chromatography step plus bioassays until ultimately we had pure individual compound or compounds that were responsible for the observed emission of light by these bacteria. So ultimately we would generate a pure compound, or in some cases there are multiple compounds that are biologically active in quorum sensing, and so I'm going to put that as pure compounds, possibly plural. From there, we would use all of those techniques that we learned a couple of modules ago for identifying organic molecules, such as 1D and 2D NMR, mass spectrometry, IR, X-ray diffraction or X-ray crystallography, I'll abbreviate that as XRD for X-ray diffraction, to identify the complete chemical structure of these molecules. In the next video, what we are going to look at is what these structures will look like. What types of compounds, by using this bioassay-guided fractionation scheme, what types of molecules result here at the end of this general pipeline.